It's long been clear that SARS CoV-2, the virus that causes COVID-19, can be transmitted through the air through small droplets and aerosols. The difference between droplets and aerosols is that aerosols are really small, less than 5 m, small enough to travel over long distances and deposit in the lower respiratory tract. Although the indirect evidence for airborne transmission is overwhelming, direct evidence has been more difficult to obtain. In a new preprint, NIH scientist Vincent Munster and his colleagues report on a new and ingenious experiment that provides direct evidence. The experiment used the Syrian hamster, Mesocricetus auratus, as an animal model for what happens when a person is infected with SARS CoV-2. Of course, hamsters and humans are different organisms, so Munster first established that both respond the same way to SARS CoV-2 exposure. The researchers used a cell culture that had been treated so that some of the cells behaved at the molecular level like hamster cells while other cells behaved like human cells. Using a biotechnology called a pseudovirus that would act toward these cells like SARS CoV-2, the authors determined that hamster cells were no more or less likely to be infected by SARS CoV-2 than human cells. In sum, this experiment showed that the Syrian hamster is probably a pretty good model for human infection with SARS CoV-2. Next, the researchers set up an experiment to see if hamsters could infect one another through the air. Cages were set up with a tube connecting the cage of an infected donor, hamster, and then the cage of an susceptible recipient, hamster. The hamsters could not move through the tubes, but airflow would allow airborne particles, if there were any, to move from the donor cage to the recipient cage. Experiments were done with connecting tubes of 3 different lengths, the longest being 2 meters. At this distance, the scientists determined that virtually no particles greater than 5 m moved from the donor to the recipient cage. Despite this, all 4 susceptible hamsters were found to have become infected. Since no large droplets could travel the entire 2 meters, the transmission must have been through aerosols. Now, for the really interesting thing, these researchers did 2 experiments with both the original SARS CoV-2 virus, which they call lineage A, and the alpha variant, which they refer to by the scientific name V.1.1.7. First, to determine if there are differences in transmission efficiency between the 2 strains, the scientists set up experiments over 2 different periods of exposure, 1 hour and 4 hours, using the 2-meter cage setup. What they found is that the alpha variant transmitted to all of the recipient cages with just 1 hour of exposure while the original strain transmitted to only half of the recipients.